Our second presentation of this morning is entitled Anatomical Characteristics Correlated to Peg Strength in Arachis Species, and our presenter is Chandler Levinson.
Thank you for the introduction, Dr. Kimmick. Is everything looking okay?
Yes.
Okay, great. So I am a plant breeding student at the University of Georgia under Dr. Peggy Azias-Akins, and I would like to start with a pop quiz that I was given in one of my plant breeding classes at UGA. And so the question is, what are the top 3 peanut breeding goals? I'm sure you know the first one is yield, but what about the second 2? And I didn't get this right, but the answers are yield and also yield. Now, I bring this up because even though I'm working with wild Arachis species known to have low yield, I'm still focusing on the fact that peanut breeders and peanut farmers want to increase their yield. So saying that, you probably want to ask me some questions. If I know they have low yield, then why even try to use them? And the answer to that is that they have strong, diverse resistances to many different types of pests that can be moved into cultivated peanut to protect the high yields that have already been achieved in cultivated peanut. Now, you might want to also ask, aren't there drawbacks to using these species? And the reason we're studying these species is so we can figure out the drawbacks and then negate any potential drawbacks that they could have. So they have small seed size, you can tell from this photo, that would decrease yield. They have trailing branching growth that makes them difficult to harvest, and they also have long pegs. But what hasn't been documented in the literature is whether or not these are weak pegs that could affect yield. So we wanted to change that through this study. But first, let's talk about what's been done. There's been 2 papers that have looked at peg strength in relation to peg anatomy. The first was in 1983. They studied strictly cultivated peanut, and they found that strong pegs had large and closely spaced bundle caps. Now, if you're looking at this image and you have no idea what it is, don't worry, we're all in the same boat. So you can think of it as a house with the roof taken off. So the peg's been cut in half and you're looking down into it. Now, there's an arrow pointing to a bundle cap. You can think of a bundle cap as a support beam in the wall of the house. So what this paper found was that a peg with large support beams that were closely spaced together made for a stronger foundation. So this makes a lot of sense if you think about it this way. What they didn't find was a relationship with peg strength and peg diameter or the number of vascular bundles per peg. The second study done in 1988 was a follow-up to the first study. They wanted to see if those observations held up with more robust statistical analysis. And what they found confirmed what was found in the first study. So they also studied just cultivated peanut, and they saw that peg strength was significantly and positively correlated with bundle cap length. So the research objectives of this study, building off the previous 2 studies, were to do this type of research, but also include wild Arachis species, and to see if there was peg strength differences between wild species and cultivated peanut. And if there were differences, then we wanted to see if there were differences in peg anatomy that could account for the differences in peg strength. And below is a cross-section of a wild species and a cultivated peanut, and you can tell from the images that we're likely going to detect differences in peg anatomy. But what about the hypotheses? We had 2 null hypotheses. The first was that there's no difference in peg strength nor in peg anatomy between Arachis hypogea or cultivated peanut and wild species. The second null hypothesis is that there's no correlation between peg strength and peg anatomy in Arachis materials. So let's get into how we tested these hypotheses. First, the plant materials that we used 2 peanut breeding lines from Dr. Colebrook's USDA peanut breeding program in Tifton, Georgia. And then we also used 4 allotetraploids. An allotetraploid is when 2 wild species are crossed together and then the DNA is doubled. This allows us to cross it to the peanut breeding lines, which we did, and we included 6 different combinations of the peanut breeding lines and the allotetraploids. Lastly, we included 7 different wild species that are diploid. The middle column of this table is the shorthand abbreviation that I'll be using of the genotype names from forward on. So how did we study peg strength? We used 5 pegs per genotype, and they had to have a pod maturity with an orange mesocarp. To study the peg strength, this isn't a real image, obviously, but we used a digital force gauge that was attached to special clamps to hold onto the peg in the pod and pull it until the peg broke. And then it was able to record the force needed. Then the peg tissue closest to the stem was excised and fixed to later look at peg anatomy. For peg anatomy, we used 3 of the 5 pegs that had the cleanest excision sites. Cross sections were taken and viewed under the microscope at 5x and 10x magnification under white light and UV light. And then we used Assess 2.0, which is actually a tool used by pathologists to study things such as lesions on leaves. But by calibrating the images in this program, I was able to treat different parts of the peg as lesions to get measurements such as area or length. The statistical analysis included ANOVA to test genotype effect on peg strength in different peg anatomy parameters, as well as Tukey's test to look at differences between the genotypes. So what were the peg anatomy parameters? First was peg area, then bundle cap area, which was actually an average of all the bundle caps per peg, total bundle cap area, and then total bundle cap area as a percentage of peg area to see if this ratio had importance, bundle cap count, the distance between bundle caps, and the total distance between bundle caps. So let's get into the results. This first table shows the ANOVA output that was testing genotype effect on peg strength and peg anatomy. In the first column is the parameter, the second, the F value, then the third column, the degrees of freedom for the numerator and denominator, and finally the P value. So in the last column of the P values, every single parameter was significant at least 0.05. So genotype did have a significant effect on peg strength and anatomy. So let's look at the specific differences between genotypes for what we measured. First is peg strength. This is a box and whiskers plot with the force needed to break the peg on the y-axis in newtons. Already shown are the 2 breeding lines. I just revealed the F1 hybrids, which were similar to the peanut breeding lines, so the peg strength was already recovered in F1s. The allotetraploids are a bit lower, with most of them being significantly lower than at least 1 peanut breeding line. And lastly, the wild species were also low compared to the cultivated peanut. But I do want to highlight Arachis valida, which was not different from either of the peanut breeding lines. And this was really surprising. So getting into peg anatomy, for the average peg area, which is on the y-axis here, and in dark blue, the peanut breeding lines are shown. The F1s were not different in peg area than the cultivated breeding lines. And for the most part, neither were the allotetraploids. But when we get into the wild Arachis species, they had smaller peg area, except for, once again, Arachis valida, which was not different from the peanut breeding lines. So talking about the average bundle cap area, which is on the y-axis in this graph, the 2 breeding lines are shown in dark blue. Again, most of the F1s are not significantly different from the peanut breeding lines, but 3 of them were at least lower than one of the peanut breeding lines. The allotetraploids were also, some of them were less than the peanut breeding lines and some were not. But again, it's the wild species that had lower bundle cap area. But once more, Arachis valida is not different from the cultivated breeding lines. So it's really sticking out. For bundle cap count, or the number of bundle caps per peg, the peanut breeding lines had about 17 per peg. And again, the F1s are not different from the peanut breeding lines. The allotetraploids were also not different. And it's the wild Arachis species that had less bundle caps per peg. For distance between the bundle caps, I went ahead and put the whole graph here because Tukey's test did not find many different groupings. So what this graph is showing is that there aren't significant differences between the distance between bundle caps for any of the genotypes for the most part. So getting into one of the most important results is looking at the pairwise comparisons of peg strength and peg anatomy parameters. I highlighted in red the most important part of this table, and that is that peg strength is highly and significantly correlated to peg area, bundle cap area, total bundle cap area, and bundle cap count. So let's talk about what these results mean. Revisiting the first null hypothesis, that there's no difference in peg strength nor anatomy between cultivated peanut and wild Arachis species, we rejected. We found that wild species had weaker pegs, smaller bundle caps, and less bundle caps when compared to cultivated peanut. The second null hypothesis was that there's no significant correlation between peg anatomy and peg strength in Arachis materials, which we also rejected. We found that peg strength was highly and positively correlated with at least 4 different parameters of peg anatomy. So let's talk about the significance. Genotype had a significant effect on peg strength. Specifically, wild peanuts and allotetraploids had weaker pegs than F1 hybrids and peanut breeding lines. So the significance of this is that peg strength was already recovered in F1 hybrids, which is an early crossing material in a back cross breeding program that we usually use with these wild species. That means that the weak peg strength of Arachis species is probably recessive and will likely be selected against in the process of introgression. So another finding was that peg strength was strongly correlated with peg area, bundle cap area, and bundle cap number. This supported the previous results found from the 2 mentioned papers earlier, that peg strength and bundle cap area or length and count are correlated. So the significance of this is that in future studies using peg anatomy to predict peg strength, bundle cap area is the best predictor of peg strength because it was significant in this study and in the previous studies. And lastly, I can't go on without mentioning Arachis valida, which was that superstar wild peanut that had a high peg strength comparable to the peanut breeding lines. The significance of this may be crazy, but it's that some Arachis species may actually have beneficial alleles for peg strength that could be used for cultivar improvement. And this is a hard pill to swallow, but this has been reported in previous studies too. One found beneficial wild Arachis alleles for seed size in a BC2 F1 population. Now you saw that images of Arachis species with small peanuts. So it might be wild, but there could be an allele that would actually, once moved into the right genetic background of cultivated peanut, can actually make a better peanut. So for future research, we found that weak peg strength is probably recessive and it'll be selected against more easily during introgression. So we don't need to worry too much about it. Since that's the case, my future research will focus less on peg strength and more on mapping QTL, a resistance QTL from these materials and moving these resistance QTLs into breeding lines that peanut breeders such as Dr. Holbrook can use to make better peanut cultivars to later share with farmers. So with that, I'd like to thank my advisor, Dr. Peggy Zeiss-Akins, her lab members that helped, and our collaborators, Dr. Gao, Dr. Holbrook, Dr. Stalker, and also our funding sources. So with that, I'll take any questions. Thank you for your attention.
Thank you very much for a very good presentation. We have time for questions. One question that comes from Dr. Barry Tillman, but also there's a couple others who've asked a very similar question, and it is this: were pegs removed from the pods before strength testing? If not, where did the peg usually break from the pod? At the union with the pod or somewhere along the peg, and did that differ among genotypes?
That's a great question, and it was similar for all genotypes. I didn't include the data, but the question's correct that it did break more closer to the pod.
So if just to follow up with that, so when we're talking about peg strength, we're talking more about the juncture where the peg pulls away from the pod as opposed to the peg itself snapping. Is that correct?
Actually, it's more of the peg snapping, but the place where it snaps is closer to the pod, but not directly on the pod.
Okay, thank you. And our next question is from Dr. Cristiani Pallone, and she says, what position in the peg did you get, from which position on the peg did you get your cross section?
Okay, so for brevity, I didn't include all the data, but I did take cross sections from the part of the peg closest to the stem as well as the part of the peg closest to the pod. That said, since one peg is an experimental unit, I had to analyze those cross sections differently, but the two data sets showed extremely similar results. So basically you got all the information just from this presentation on both peg areas of where the peg was taken.
Thank you very much. That's all the time we have for questions, and thank you for your presentation.
Thank you.